Oh, I see you.
Oh. You got 4B filled in about the reduced phospholipids.
Yes.
Let's move on then. And so we got to stop by 1:45. There's new time missing that. I don't mind the hour extra. Okay, here we go. There's 5. Oh, now before I start, now we're talking about the rough ER. Okay, it's the rough ER. Now, there we go.
Were you finished with it?
Yeah.
And I'll say some things about these in a minute when you get through getting the blanks filled in. And I'll start putting together that paper test for lessons 1 through 8.
When's that test?
Monday?
I don't get any bigger than, I can get bigger and bigger and be a heck of a test. I figure I better stop now. I wish I'd done lessons 1 through 4 and then 6 through 8.
You could
I was
always do that
rolling.
anyway.
Huh?
You could always do that anyway.
It's over how many chapters? I mean, how many
8
lessons?
lessons.
8 lessons?
And if I change my mind, I'll announce it. If I do the first 4, if it gets to be like 1,000 questions, I ain't doing that. But if I can cut it in say the first 4 lessons, and I decide to, I will announce it. I'll be watching Angel for a word from me about how I'm doing that test. I won't know probably until I'll start working tonight on it. Barring I have any papers to grade from the class and behind y'all. All right, let me explain what's going on with the first one. Okay. I know you've made your lunch for, right? You made your sandwiches. What's the last thing you do at a sandwich before you put in your lunch bag?
Put it together.
That sandwich baggie?
You put it in saran wrap or a baggie. Okay, you package it. What you're going to find happening here is that the rough ER, it makes the sandwich. It's just bread and ham. And then the lysosomes and the Golgi apparatus, they take that sandwich and they wrap it like in saran wrap. They package it. Then they send it out. So it's a teamwork here. Like if in my house, if I'm making lunch like I do sometimes for my wife, I would make the stuff and give it to her, and then she would put it in little baggies and she'd be done. So I would be in the rough ER in that case. I'm the one actually making the sandwich. All she is doing is packaging it. And that's what the last 2 things do. Now, the white blood cell. The rough ER is a tunnel that connects to the outside of the cell. That's the entry point for germs. They can crawl in there and crawl right down that sewer line. Well, they contain white blood cells, which can kill what's inside there. Yes.
On point 6, digest needs to be read in that 2 left.
Oh, swap those 2?
Yes.
All right. Thank you. ER. And it's smooth because it ain't got all the bumps. The bumps are the ribosomes. They're missing, so they're not making that much stuff. They do produce membranes for the cell. They produce more cell membrane because the smooth ER is pretty far out there. It's a long way from the nucleus. It's almost at the cell membrane. So it makes sense that this structure is making more cell membrane as the cell grows. They produce steroids. Like in a lady's ovary, that's where you find her estrogen being manufactured in her smooth ER. Well, I mentioned that. Ovarian follicles of a woman's ovary, the smooth ER in those follicles are making the eggs. Eggs are called ova, right? Ovary is from the fact that it makes eggs, which is called ova. The name makes sense. In the case of the man, they're called interstitial cells, and they're busy making testosterone. But where it's being made at is in the smooth ER in his interstitial cells, in his smooth ER. And that could be true for the thyroid gland, where it's making the thyroid hormones. It's true for the adrenal gland, where it's making adrenaline and norepinephrine, things like that. So many of your hormones are made by the smooth ER inside a cell. Cell structures, more of that cytoskeleton, that's being made by the rough ER. That's a lot of proteins. And they use, it makes sense that if you're going to make the membrane, you better make the fat too, right? Because the membrane is so much fat. Well, these things can synthesize fats. And then use those fats as it makes membrane. Because for a membrane to be effective, it's full of fat. That's what keeps the water out. Now, we're talking about the smooth ER now. Are y'all ready? I guess so. Okay. There's a lot of it in the cells of the liver and cells of the kidneys. There's a lot of smooth ER because that's where those two organs, actually 3 organs, if you count 2 kidneys, they're busy destroying poisons. And one of the roles that the smooth ER has is to detoxify drugs. And since the liver is a major detoxifier, it's full of it, and your kidneys clean your blood, they're full of the smooth ER. You won't find it in the stomach. The stomach's not cleaning anything, it's digesting. But because the liver and the kidneys play a major role in taking out chemicals that would hurt you, but they have a lot of smooth ER. Calcium is stored in the smooth ER, especially in skeletal muscle and cardiac muscle. It makes sense the bones have a lot of calcium. But did you know that the heart does too? A lot of calcium. Because muscles will not function without calcium. Well, the heart never stops functioning. It needs a constant supply of calcium to make it beat. I mean, this bicep, it ain't working right now. The heart is. So, cardiac muscle tissue, because in fact, even when you're sleeping, it ain't resting. It does slow down stuff. Like if you've been running a race, you might stop and walk around, right? You didn't stop, but that's easier task, is it not? So when you're sleeping, a lot of folks say, I don't need much sleep. Now, everyone needs different amounts of sleep. But even though, I had to tell my wife like that, I said, you get into bed, and because your eyes are open, you get back up and you do crossword puzzles. If you just lay there, honey, you'll rest just laying there. But she's thinking about, well, I got to do clothes, and I got to wash, I got to get them out and wash and put them in the dryer. So she gets up and does more work. And she comes to bed like about 3 o'clock in the night, in the morning. And then when I get up at 8 o'clock, she's like a baby gets your days backwards. If you let that little baby sleep all day long, guess what's going to happen?
It's going to torture you.
He's going to be up all night long. And you're up all the time. Because when he's sleeping, you're catching housework. And when he's awake, you take care of him. So when Mama get rest?
Not when Dad.
That's when you learn to keep that little rascal up in the daytime when you're up. Don't let him take those naps like you like to do. Because they're going to sleep regardless. But a lot of folks say, get his down. I can rest for a minute. So you let him sleep all day long. At nighttime, buddy, he wakes up ready to go. And you're going, I can't keep doing this. And then you soon learn to keep that baby up in the daytime when you're up. Don't let that baby sleep.
But
The nap is fine.
He'll get you right.
you have to judge that too, because
Oh,
you
sure.
can spend too much time trying to keep that baby awake and you didn't wear yourself out. So,
You got to find that perfect medium.
Yeah.
And usually the first baby, you don't find it. The second baby, you go, I got you, I got your number.
And what if you have twins?
First time or second time?
No, like what if you have twins your first time?
Then you have a lot of learning.
Or triplets.
It's going to be an experience.
Yeah.
I would rather have twins second time, I think, if I had to have twins at all. That first baby, I got to admit, when Robbie out of the baby, we thought he was porcelain glass. You could break him. we treated him so carefully. And Tim, my second child, we threw him around the house. We learned from Robbie that he's not going to break. And if he's crying, and he's not wet, and he's not muddy, and he's not hungry, let him cry. He's crying for one reason, get your attention. And they're good at that too. It's amazing. How can a little kid who has no book learning master you so fast? They sure can do it though. They can make you jump through hoops, and that rascal can't even talk yet. This is a picture I found. I ain't sure where I got it from. I liked it. Of them, rough ER and smooth ER together. And you see all the little dots. And those are ribosomes. Those are ribosomes. And when you see the dots, we call that the rough ER. And when you don't see the dots, like out here, we call that the smooth ER. They're doing different things. It is like a tunnel, though. It's like a sewer system where you can get in the sewer system and crawl all around the city, kind of like that. Or a subway, where you go from here to there underground. But that's the ER, and I guess it has one function. I would say transportation. They manufacture those proteins and they ship them out. They transport them to where they got to go. The next picture, oh, I just got some more to do, don't I? I didn't realize that. The one on top is taken through an electron microscope. The one on bottom is, try to draw it. Thank you. This is the tunnel. And can you see a dot there? And a dot there. We can draw them really pretty. But when you take the scope out and take a look at them, they never look as pretty as we drew them. And if you look, do you see how long and flowing these membranes are? See how kind of roundish these are? You see it there? Now, it's a pretty good job. I got the long flowing membranes of the rough ER, and I got these little circling segments. This does not look at all like that. Would you agree? Well, this is the smooth ER. And if you notice, there's no ribosomes on the surface of them. And they even, when you cut them in half, they even look different. So the artist, I thought, did a pretty good job on his drawing of the one on top. A lot of times, if you look at the drawing and look at the real picture, you start to see what he's showing you.
What's the big part right on the top one there looks like a little lake?
I mean, like that little lake right there?
Yeah.
Those are vacuoles. Might be a lysosome. Those are internal organelles. It could be a lysosome. There's also things called peroxisomes in it too, which look alike. Yes.
What was that C word? Can you go back one?
The C
One
word.
slide.
What C word?
Cisternate? Cisternate. Okay.
Oh, cisternae. C-I-S-T-E-R-N-A-E. That's the word used around the board. It's not just for this thing. Cisternae are found in a number of organelles, just like saying folds. We just say cisternae. The ER has cisternae. The thing called the Golgi body has cisternae. Inside the mitochondrion has those folds, cisternae. It's just a common science word. Okay? Now the ribosomes, they're found basically in 3 places. And a ribosome is made of some proteins mixed with RNA. Not DNA, RNA. And the three places is A, B, and C in there in that second and third line is where you find ribosomes. You find ribosomes in nucleoli, which means plural, right? You find them floating around, just floating around like leaves in a lake. And the cytosol, which is also called cytoplasm. And you also find them, like you know from previous slide, hooked to the rough ER. That's why it's called rough for. Now number 10 is going to tell you what it's doing. The ribosome gets his orders from DNA. The middleman who brings the orders is messenger RNA. If I am the DNA and you are the ribosome and Haley is the messenger, I give her my orders, she gives them to you, and you do what I want you to do. That's how it works. And the only thing that ribosomes can make are proteins. They can't make fats, they cannot make carbs, they only make proteins by hooking together amino acids. Amino acids are the building blocks of proteins. Like my house is made of bricks. My bricks are my building block. Well, in the case of a protein, you hook those acids together and they start to wrap around themselves and become very tangled up. But if you untangle them, it looks like a pearl necklace, all wadded up together. Proteins are very complicated because if you took and strung, let's say you're going to string a 3-foot long pearl necklace, that's long, right? And you're going to wad it all up and lay it here. Could you follow it by just looking at it? You have to unwind it, wouldn't you, to see where it all went. Well, but the protein is all balled up like that. If you unwind it, you'll start seeing, oh, that's just one long train of pearls. I didn't know that. When you first look at it, you go, like a huge mess. And they are. We all know that if you want to go a long time between breakfast and dinner, for breakfast, eat meat product. Meat products are full of protein. It takes a long time to break it down. A bowl of Rice Krispies is good for the time you eat it. But about 2 hours later, it's gone. And you're going, 11 o'clock, 12 o'clock, I'm hungry. But if you have scrambled eggs in the morning or maybe a sausage biscuit, you'll find by 11 o'clock, you're still not hungry because that's still in your stomach. And that's how a lot of those use protein in their diet. Because if you're not hungry, you might not, but you're not going to be eating a whole lot all the time. But I like a bowl of, cornflakes. It's always good and refreshing. Don't stay with you though. It's gone. Scrambling eggs to me is a little harder than just pouring milk into a bowl of flakes. But the scrambled eggs, if I told my kids in high school, some had a lunch period start at 1 o'clock. But if I'm used to eating at 11, I can't say, well, for breakfast, eat you some scrambled eggs, a lot of them don't eat anything. They come to school and by 12 o'clock, and I'm trying to teach this, they're thinking about hamburgers. And that's why I tell them, you'll be much better off if you eat you something in the morning. That will prolong your appetite to about 1 o'clock. You probably can't eat. But I'm the DNA, I got the code. I know what our proteins I'm going to make. But I can't talk to her. But Haley, I can talk to her. So I tell Haley, you remember how you do the up and the down? Have you heard that before? Where in the code, adenine always pairs with thymine. Don't write me that. Don't write that. I'm just talking. And cytosine pairs with guanine. They're opposite. What's the opposite color of black?
White.
Okay. You always work opposites. If I want a black pearl necklace, I will tell her white. What will you tell her? Black. She'll lie. I know she's gonna lie. So if I want a black necklace, what am I gonna tell her?
White.
And then she'll lie to her, and I'll get what I want.
So you start with the lie first.
Oh yeah, I lie to her.
Then why does she change your line to a different line?
Then you have melanoma. You have skin cancer. Some kind of disease came from the genes. breast cancer, runs the family, genetics. So if I want something pointing up, I'll tell her,
Down.
and she'll lie to her,
Up.
and I'll get what I wanted. That's how it works. So, and I see, adenosine's partner is thymine. If I want adenosine, I will tell her thymine. No, she won't lie to her and tell her what? Adenosine, which is what I wanted. I trick her.
Okay.
That's how it works. And we don't understand why it does it that way. We know it does though.
But sometimes
It's called complementary basis. And I have a little code I remember. It's a little sentence. It's the letter A and the word at T. Because A is always found hooked to T.
Adenosine.
So my little code is A at T.
A
And
at
then if I know A is always hooked to T, then the other two, C and G, then they're always partners.
What if you?
And the reason is, there's a wall plug with only 2 holes in it. Could you put a 3-pronger in it?
No.
No. Well, it works this way. Adenine has 2 plugs. So does thymine. But cytosine's got 3. And so does guanine. There's no way you're going to make a 3-pronger hook up to a 2-pronger. And that's why A never hooks to C or G. It just won't happen because it's like plugging a wall in. And the opposite says, if I want A and the opposite is T, I'll tell her T. Knowing full well she's going to tell her A. Which is what I
but she'll
want.
give you the right. She'll give you the right thing.
Based on what I told her.
Okay.
Because she's going to always flip it over. She's going to always flip it over on purpose.
Okay.
And so if I wanted C, I would tell her G. Knowing what she's going to do is tell her C, which I tricked her. We call that what, reverse psychology? We do kids the same way. We tell the kid, you don't want that gun, do you? Or opposites is always, that's how this works. And it all boils down to the amino acids you get. Because if I want that black pearl necklace, she's got to get the black amino acids to make my black pearls. So if I want black, I'll tell her white. She'll tell her black, and she'll work herself to death, making me my black necklace. Now I wondered, but now if I told her black, it'd be wrong. So it's got to be done opposite. And I got a PowerPoint coming which explains all that, and we're taking little small pieces.
Who figured that out?
Now, huh?
Who figured that out?
Washington Crick and Rosalind Franklin and Maurice Wilkins. They figured it out back in. As a matter of fact, I was born in 50. They figured it out in 52. I didn't know about it. 2-year-old kid, I could care less. But they won the Nobel Prize in 56 or 58 for having done that. But Rosalind Franklin was the lady who died before the prize. She had ovarian
I've met
cancer.
Maurice.
She died.
Met him.
Watson didn't like her.
Yeah.
She thought
Right.
her
Up
place
here
was in
on
the
your
house, pregnant and in the kitchen.
Got it.
Thank you.
But matter of
Thank
fact,
you.
she took a picture, and that picture was gotten by her boss, Maurice Wilkins. And without her consent, he gave that picture to Watson. Watson saw the picture and goes, "That's it." He took that picture, and then from that picture, he figured out the structure of DNA. And never one time did he tell her.
They wrote the best
Like he did his
chemistry
hand.
book.
And in that, he wrote a book. I read the book. In this book, you think he was the world's best scientist. He had ego. And he called her Rosie. Because she didn't like Rosie. Her name was Rosalind. He referred to her as Rosie for one reason, to get her going. And at one time, they said, Maurice Wilkins walked into office. She about ready to punch him out. She was so mad at him. I think what happened, she discovered he had a picture. And he wrote the book. He got the money. He got the prize. Now, if she had been living, it'd been better, but since she died, and you cannot, no one can win a prize after your death. But she had been living, and they have now got a new book out. Rosalind Franklin Woman of Light, something like that. But the writer really tears Watson up. Crick, his partner, liked her. And when Crick read Watson's book, he wouldn't give a forward. He wouldn't write anything about it. So Watson said to James Crick, "Here, read this for me and tell me how good I am." And you write a little forward like books do, you know. And Maurice Wilkins, he wouldn't do it either. He said, "This ain't right. This ain't Rosalind. You got to paint it as a bitch." He did too, baby. And when the book is over, so she's dead. And the last thing he says is that of all the things I wrote about in here, I got to apologize to one person, Rosalind Franklin. I did her wrong. I didn't realize how much trouble she had as a woman in a men's world. He never said, "I'm sorry for taking your picture," though.
Many men don't say
And
that.
Rosalind, there's a movie out. It'll make you so mad. And you look at Watson. He looks like a little nerd. He didn't really like that. There's all kinky and curly.
What's
He was
the name?
smart though. He was really a smart man.
I've tried to look it up. There's like a Jeff Goldblum movie or something.
A what?
A Jeff Goldblum stars in the movie. It's called like Life Story or something
Yeah,
like that.
something like that.
Okay.
I got it from Netflix and I watched it.
Oh, really?
Go on Netflix and just type in Rosalind Franklin. You might get it that way.
Was it like a documentary style, like educational?
Half and half. Because Maurice Wilkins, the real man, is in the movie talking about Rosie.
Okay.
But Rosie's dead; she can't talk. An actress played a role. I thought very. I knew the history, so I felt good about the movie. But it really paints Watson in a bad light. He's still living today, by the way. He stopped doing speeches because he did. First off, he's a bigot. He's a racist. He hates Jews. He's smart. And people have stopped inviting him to do speeches, and he's bitter about that. He's still living for it. Unless he died yesterday, he's still alive now. The only one left is him, the man who wrote the book, James Watson. But that's a truly sad time in science when that woman, Rosalind Franklin, who did so much, got no credit whatsoever. My wife, I showed her, she got spitting mad. I said, "That's the way it goes. Back in the 50s, that was. That's how it was. It wouldn't fly now in 2010 or 11, but back then, women's place was not in the laboratory. It was in the kitchen. And you know, she didn't do anything. She didn't do anything about it. Even when she was living, she didn't say anything. And the first time Washington showed her his model, she laughed at him. She said, "That's so stupid." I made him mad. He didn't ask her to come back again. Because in DNA, do you remember the bases are inside? Do you know that, right? They're inside. He had them outside, sticking out.
Connecting to nothing?
To nothing. And she goes, "How's it going to stay together?" Because we now know that you have the rungs on the, sugar, phosphorus. He knew that, sugar, phosphorus. And he knew about the bases. And we now know they're inside, hooking each other like a ladder, right? But when he first showed her his model, which is made of metal, his bases are on the outside, and even I'm going, "There's no way that can work." And she said, she flat out, "That's stupid." And because he never offered to let her look at one again till it was finished. When they finally got finished, he let her see it. And she goes, "That's it." But she was still seething because he would never have got it if you're not seeing her picture she made, which her boss gave to Watson without her knowledge, and it's just terrible. But that's one. There are a few other cases where people have been not given their just due, and Rosalind Franklin probably is top of the heap. That's the most unjustly treated scientist ever. When she went to school as a professor, a doctor, she couldn't even sit with men. She couldn't sit with them. So she had no one to bounce her views off of. Well, all the guys sat together and discussed this, discussed that, so that won't work. So actually, she's even better because she did it all herself. But no one did. even in class, we talked to each other, right? She had no one to talk to. And she did it all by herself. That woman must have been really, really smart. Really, really smart. Golgi Complex is named after a man. That's why it's always a capital G in the front. You never want to see a small G there. That's the guy's name. Emilio, I forgot his first name now, but these have cisternae, and there's a word again for you. I think Melissa asked that. Who would have said that, Melissa? Okay. They have cisternae, and they're flattened membranes. But you remember that the ER had all ran together? These are separate stacks, like pancake stacks. They're not connected to each other. And you'll see a picture, you know what I mean in a minute. They produce the carbs. They make the carbohydrates. They also can package the proteins that came from the ribosomes. They also can package the glycoprotein. Glycoprotein is a protein that's kind of mixed with carbohydrate. Because glyco means carb, as in glucose. In the very beginning, glucose was called glycose. And we changed the spelling to GLU. It used to be GLY. So that's why some of the words are cut. Yes, sir. I'll save it. It'll be done next time. Folks think I'm real smart. Have y'all proofread it for me. Okay, the Golgi apparatus, well, I said apparatus. Some folks call them Golgi bodies. I've heard them called Golgi Complex. I've heard them called Golgi apparatus. It's always Golgi something. This guy's an Italian. All right. The lysosomes, they contain enzymes that digest. And the enzymes are made by those enzymes are proteins. They're made by the ribosomes, which are stuck onto the rough ER. The word secretory means to put out or to release. And the pancreas releases insulin because the pancreas, the cells of the pancreas, the protein is made by the Golgi, by the rough ER. Like me making that sandwich. It's got to be in a bag, right? Well, then they send the rough products to the Golgi apparatus, where the Golgi wraps it up and sends it out. And that's the insulin. All your glands, all of them, have these secretory vesicles. That's how they produce their hormone to kick it out there, whether it's adrenaline or whether it's estrogen or testosterone. They all have the same mechanism. Now, if you look, you see cis, you see medial, you see endo, you see trans. Those are the four regions of the cisternae stack. Cis. I remember CMET. CMET. And that's how I remember those. The luck is, well, a little acronym. CMET. Yes. Okay. Thank you. And I've listed them in order that the material passes through. The cis gets it first, works it over, gets it just right, and the trans spits it out finished. That's the order it passes through the Golgi apparatus. And you'll see a picture in a minute showing you how this would work. So something comes from the ER and attaches to the cis, the first front of the Golgi. And then that works through the medial, through the endo, and finally through and out the trans And that's when your sandwich has been packaged and your lunch bag and you're on your way. Okay? And like I said, the trans, that's where it finishes. As in transport, yes. All right. Each region, the cement, the cyst, the medial, they all have their own enzymes. And one after another, these things are hit as they pass through. And they modify, like, how many folks trim the crust off your bread? My wife does. She trims it off. Well, that would be a modification, wouldn't it? So they'd be trimming crust off as it passes through. When it gets done, the sandwich I gave her might not look like the one I made. It might be sliced in half like she likes. The crust might be cut off. But all that modification to slicing it and removing the crust and whatever is done by the Golgi. I just make the sandwich, crust and all. I don't cut it either. Going to be cut it?
Yes.
The Golgi got to cut it for you. I'm not cutting it. I'm just going to make a sandwich. And actually, that's how I did my household. I would get up early and make our lunches, and my wife would before dinner. And I knew not to put in that bag of it. She was going to cut it up and slice it. I didn't like putting those two small pieces in the bag because they all fall apart. I'm going, why don't you just leave it alone? That's a woman for you. I got to figure it out. I had no idea why she cuts it up. But I know she likes it cut up. Matter of fact, I don't want mine cut up. Go cut mine up. I want to eat it. Now there's a picture two ways. There's an artist rendition, and then there's the actual electron microscope. And you see, they look very close to each other. Do you see the curvy nature right here? That's right there. Now look carefully. This never does touch this one. They always are separate. That's not the way the ER is. They remain totally separate. This is the cis side that's receiving things. That's the trans side that's kicking things out. Now, I see it good here. I don't see it that well here. But even here, they still remain. That looks like 4 stacks of bologna. Follow me? 4 pieces of bologna kind of wrapped around. This is the side receiving. You see this? You see those? You see those? You see those? They're coming from this way. They're coming from the rough ER. They're going to pass through this, and what comes out is a finished product. Much different than what went in. Well, I give that sandwich to my wife. What's in that bag and nowhere near what I made for her. Here's another picture. On this time, we got this side is the cyst. That is some of the ER right there. That's a vesicle traveling this way. Now see all the ones being put out over here? That's going away. Cis, M, E, T. And the trans space is over here. And all these are the bagged sandwiches that came in this direction. And here it's been chopped in half as the crust is cut off of it. Totally different what went in.
So where does it enter in the E? Are you saying that's?
It enters on this side. So when I get it made, I give it to her, and then she works it up, puts it in a sandwich bag and her lunch box, and this is the finished product over here, the trans space. The various cisterns, they're called faces. They're not always four. They're not always four now.
Say that again. Did not.
1, 2, 3, 4, 5, 6, 7, 8. I just see 4. I think I see more than 4 here though. I'm trying to guess. 1, 2, 3, 4, 5. That might be, I'm not sure what that is. That might be a tad of the ER. Because obviously this doesn't look just like that. But anyway, if you go back to ER, when I first showed you this, see how they all kind of run together? They're not separate stacks, are they? Well, and the Golgi, they're actually separate. They don't run together. As a matter of fact, when we first saw the Golgi, some folks thought there was some more of the ER. And then Golgi was the one, the scientist said, I don't think they're connected. I think they're separate. I don't think they're a part of the ER. And when further research found him to be true, we named him for him. He didn't name it himself. We named it Golgi in honor of the man who first got us thinking about it, that that's not more ER in there. That's something different. Always found in the nucleus, never out there toward the cell membrane. It's always close to the nucleus where things are coming from. Because I'm in the nucleus, I'm the boss. Haley, the messenger, is leaving and going to talk to her. She's in the rough ER, close to the nucleus. Now Stuart might be the Golgi, and when she gets it done, she gives it to him. And he packages it. And he sends it out to the rest of us. It's a conveyor belt type effect. The lysosomes are another kind of organelle you find inside a cell. Now these things can be dangerous to the cell if they're not handled properly. They contain enzymes which digest things. They can even digest the cell itself if it were to break. So far we have identified 50 different enzymes in these lysosomes, all doing different things. Remember now, enzymes are specific. Each one does a special job. None of them do 4 or 5 jobs. This does this, that does that, that does that. There's no enzyme that does 2 things. That's a lock and key thing. Make one key, one lock. That's how it works. Now, the enzymes that you find inside the lysosomes, who makes them? The rough ER. The lysosomes just bad. Just bad. They didn't make them. And guess who put those enzymes in the lysosome? Golgi did. So the ER makes the enzyme, sends it to Stuart, the Golgi. Stuart then packages in the membrane, and we call that a lysosome. He sends it out in the cell. So lysosomes came from the Golgi apparatus, who got their enzymes from the ribosome, who got the directions from, who got them from, I still control everything. The nucleus still controls everything, even the lysosome. Because what's in the lysosome, ultimately, I had to say so. I said up. She said, she went with up. Then she packaged it, then he packaged it, and I got a bunch of lysosomes filled with enzyme called up. Which one I wanted. Look how it works. Now here are the functions for the lysosomes. Lysosomes, they're very, very, they're like kind of nitroglycerin, but not drop it. Don't want to break it. Now, if you break it, outflows these enzymes that would digest the cell from the inside out. Okay. What's intra mean?
Inside.
Inside. Large molecules that get inside the cell through active transport, if they got to get rid of them, the lysosome does it. The lysosome dissolves it with its enzymes. What happens to if you walk along here and you hit that table corner and you bruise yourself? Well, in the cells that are bruised, there are cells which are damaged, organelles busted. How do you erase those organelles? Lysosome does. If you have something that's dead and got to get rid of it, the lysosome does it for you. Now, since you're erasing your own structures that are now broken, but they're yours, we call that autophagy. Now, the one on top up there, that large thing got in there. Did you make it? No. That's not autophagy. I mean, when the mitochondria has stopped its function and now just taking up place, I need a new mitochondria, but I got to get him gone first. Well, the lysosome comes up and digests the mitochondria that's broken, and then you make new mitochondria from another mitochondria. You've got to have a way to get rid of it. You can't have garbage thing around your house, you might. But cells are very clean. And autolysis, auto means self. The key word is apoptosis. Cells die. And new cells arise in their place. The dead cell has got to be gotten rid of, or you get gangrene like I got. And since they couldn't get rid of them, they took off my leg. But in your body right now, cells are living, cells are dying. Cells are dying, cells are living. And those dead cells have got to be get. This dandruff, we get rid of that way. But inside your body, you can't do that. You can't get rid of them by sloughing them off in a wash rag or in your shower.
So when it does that, is that like how we saw the other slide, where it comes and circles it and then releases it?
Exactly.
So then what happens when it's released? It goes in what, urine or something?
Pinocytosis kicks it out, urine picks it up, gets rid of it.
Okay.
And if it's poison, it first goes to the liver to be depoisoned. That's why alcohol attacks your liver. And when the liver stops to function, you can no longer detoxify poisons and you die of liver failure. Actually, you poison yourself to death because your liver has stopped working for you. Because alcohol kills all cells, including liver cells. It's called cirrhosis. Now, I don't mean a person who drinks a mixed drink at Thanksgiving. I mean a person who drinks it every day. The social drinkers are not ones that are in danger. But you got some folks that drink liquor like I drink water. Well, like I drink Coke. Coke ain't good for you either, to tell you the truth. Water's the best thing for you, but water's no fun to drink. It don't burn going down like a Coca-Cola does. All right? Glycogen now. The liver takes this extra glucose you have and stores it as a thing called glycogen. And when you want the glucose back, it must tear off and send it out. Well, the lysosome allows the liver to break off the bonds and release the glucose. So between meals, you're not starving to death. That's all controlled by insulin, though. How fast the liver does that is insulin. Now, if you don't have much insulin, then you're going to have a lot more glucose being poured out, and your blood sugar levels rise, and that's the whole problem being diabetic. Now, there's two kinds of diabetes, though. There's one or two. On one of them, your liver is not making enough insulin. On the other one, you're making insulin, and your cells are ignoring it. Two medications work on both of them. On one medication, for type 1, I need to kick my pancreas in the butt, make it make more insulin. Or if I have type 2, I'm making insulin. I got to tell my cells to pay mind to it, do what it says do. Because you got two kinds. Both of them result in high sugar levels. But the medication for diabetes 1 does not work on diabetes 2 because they have different causes, different working point on there. All right? Peroxisomes are nucleon block. When I was in school, we didn't know about these things. I'd never heard of them. I think maybe because they look like lysosomes, that's what we thought they were probably. We didn't know there was another critter. Peroxisomes, they're not made by the Golgi. They come right from the smooth ER, the membrane. Okay? I didn't picture on there, but that's how it works. They're in all your cells, but again, the liver and kidneys have a lot of peroxisomes. And a free radical is what causes cancer. And these things can kill free radicals. They can break down alcohol. So they're actually, for the heavy drinker, that's his friend right there. But if you drink too much, even they can't help you. For the occasional drinker, that's what's going to save you. But for the person who walks around in their hand all the time, the wino, so to speak, that they can't keep up with that much alcohol. So these are good for us. But they make something that's bad for us. What does peroxisomes sound like to y'all?
Peroxide.
Peroxide. That's how I got his name. They use oxygen to break down organic molecules. But in doing that, they produce hydrogen peroxide, which is poisonous. They have solved one problem, but they created another one. We all do that, I think, a lot of times. You fix one thing and fixing that, the same other breaks. It's always something. But we can handle it. If you pour peroxide on a wound, what's it do? You know what, Bubba? Is it killing germs or enzymes that are activated? It's enzymes. It ain't germs, it's enzymes. All that bubbling you see, oh, kill them all, kill them all. It ain't germs. The bubbling effect is the act. If you take that peroxide and you leave the lid off because you forgot about it, and in a few days you come back, what's happened to it? It's turned to water.
Oh, really?
You see the H2O2? The lid off, that one of the O's comes off. When you lose the O from H2O2, what do you have? H2O. The lid must stay on it to keep it that way. But I've been known for, like my son got cut, so I'm so busy, I don't think I'm going to put, I'm working with him, right? And I come back later that day and go, oh my God, I quickly put it on. That's just going to make my peroxide run out faster. Are you pouring on, nothing happens, that's water.
I have a question. When it turns to water, is it like straight pure water, or is there remnants of peroxide?
Well, it depends on how long it's been sitting there.
How long does it take?
It don't change to all water. It takes time for it. The stuff on top, the oxygen leaves, right?
Right.
It's just I don't know how long it takes to do it. I wouldn't drink it though. I mean, if I were on a desert island, I might then drink it after being out there a long time. But I wouldn't walk in there and take a sip, assuming it's water.
Right.
I'd get from sink itself. Yes.
Why does it have to stay in the brown bottle?
Light will also, light hitting it heats things up. That will increase the rate of deoxification. The brown bottle just slows things down. But if you're smart, you'll cap it the moment you get done using it. Because it won't last long if you don't. Kind of like alcohol evaporates real fast out of the bottle. Alcohol evaporates. You put alcohol in your body, it'll feel cool. It's evaporating. That's what's cool for.
Is that why the alcohol companies put alcohol in brown bottles?
Exactly why.
So what's happening?
Why not? Now, rubbing alcohol in the white bottle.
No, drinking alcohol.
What's happening with the bacteria? I mean, when you get that cut.
The brown
Why now?
When you get that cut and you put the peroxide on it, of course I thought it was killing the bacteria, so what's happening to the
The enzymes are the ones that kill it. It stimulates the enzymes. You know how you take an apple and you cut it, but you don't eat it and it turns brown right there? That's enzymes too. Nothing wrong with the apple, it just don't look pretty.
What about pumpkins? I've always wondered.
Look that up. I can't keep going there. Good conversation, but let's get back on this. If you want something, then get a keyword and look it up and see what it finds for you. Now, the peroxide that you have been made, it can now break down other organic compounds. So you made this poison that's going to in fact break down other poisons for you. But you still got to figure out, I've got to get rid of that peroxide in the end, right? Well, let's get that worked out. They can use the enzyme called catalase. And what do you think? Catabolic, right? Catabolic means what?
Break them.
You break it down. So what happens inside when the peroxisome is through with the peroxide, in comes enzyme called catalase and takes all the leftover peroxide and turns it to two things: water and oxygen, which are harmless to us. And in that bottle that you have on your sink of peroxide, that's what's happening. But the catalase makes it happen real fast. On my sinks, it happens real slow. I often wonder if I could find me some catalase and put it in the bottle, see what happens when it foams upon it. I don't know. But I know if you sit it out. Now, I'll tell you one thing now. I've set that bottle out for a few days and I cap it, and it's still bubbling. I mean, it's getting weaker. What would like be fun to do is take them and buy you a bottle of peroxide and keep it always capped. Take another one and uncap it late for 4 or 5 days. And then get some raw meat. Don't cut yourself, just get raw meat. Going on there. And see if you see a difference in the bubbling effect. I've never done that before. I wonder if you'll see, I would think you would. If you pour water, then what's going to happen? Nothing. Peroxide can make it bubble. They put peroxide in here. Every year. That sounds weird, doesn't it? My wife did it with me. I don't like it. She'll put peroxide in your ear. And you hear it.
Yeah, it tickles really bad. That's not weird.
I don't like it, so I prefer her not do it to me. She does it to herself all the time.
Why?
Like she cleans her ears.
She thinks?
It might be lifting stuff up. Making them racket be doing something in there.
Yeah.
If you've not done that, it's experience. It won't hurt you. Now these are pictures through electron scope. I don't have an artist rendition. Now we're talking extremely small. Now there's some of the smooth ER. There's some more of it. There's some of it. Now, these are your peroxisomes. Now, these are lysosomes. Which one to you appears to be more dense? The lysosome or the peroxisome? And that's how we figure out. Because I'm thinking this one right here, that might be a peroxisome. You see it's not quite as dense as these are? I'm thinking these two might be peroxisomes. So could you see someone thinking they're all the same thing? Back, 50 years ago, thinking they're all lysosomes? And you start going, wait a minute, Jim, wait. You say, ain't that a little shade lighter? Aren't these two shades lighter? Like these are? Do you see a dark mass in these? I see something there. I see something there. Now these things right here are mitochondria. They got lines going across them. That's mitochondria. It's a mitochondria too, but shaped in another direction. But I can see how they would call all this stuff lysosomes and not realize that these three things are not lysosomes. Those three things are in fact these bad boys. Of course, you can always see it now. Back then they didn't know that. I understand they didn't know it. And as science marches on, we discover new stuff that we didn't know we had. When I got my degree in biology back in the 70s, all this stuff here, we didn't read this stuff. And now years later, and I'm teaching it, and I'm rereading all this stuff, I'm going, they discovered a whole lot of stuff in the time I was out of school. So a person who gets a degree in biology now has more stuff to learn than I did when I was in school. Does that make sense though? I had a parent conference one time, a lady came in with her daughter. And this lady, she's a doctor. And the daughter is struggling in biology. And the comment the lady made said, I don't understand why she is having such a hard time with biology. I said, well, doc, compare the size of your book to her book. A lot more stuff to learn. Are you comparing apples and oranges, doctor? And what she's learning now, I would dare say you didn't even know existed in your class of biology. And she goes, what do you think about that? She kind of backed off some. And when she backed off, the girl did better. It's not like the girl didn't do good under pressure. Some folks were good under pressure, and some folks don't. You got to find your niche. I work better under pressure. I never start an assignment until it's almost too late. I gotta be a little scared. If I start too soon, I'll start over 10 times. Now I'm wasting myself. So I said, don't you start yet. Go hunting, go fishing. Think about it while you're doing a reel. I think I'll do this. And boom. But see, I'm thinking when I'm out there, and when I come back, I'm ready to get something going then. But I never start. I never start. But the trick is, you need to learn how long you can wait and start till finish. And both my sons, I ain't should do it now. They would tell me the night of the project, I need a triboard. What did you know about this? About 2 weeks ago. Why didn't you tell me? I didn't think about it. Now I got to go out and pick the bushes, and I found one drugstore open, and they ain't got any triboards. And I get mad. Oh yeah, it cost you 3 times. But we all do that. We're all guilty of it, and the problem is you got to learn how far to put it off, and you still get it done. And but my sons, they had my not rage, but my discontent. Because when you get to college, I ain't there to buy the tripod for you, and you wait too late, you might get done. Some teachers don't take prisoners. They don't give you extended time frames. All right. Well, let me see what's coming next. Good. We're on a brand new topic. We'll start mitochondria when I see you Monday. Oh, you both came in late?